your cameras on, and that is it. Okay, I'll go over a little bit of what to expect on the exam later on. Any questions? Okay.
Will we have a study review for the exam, or is it just I don't know if we took it so far?
There's a study guide up also.
Okay. Okay. Thank you.
And I'll tell you what to expect also.
Thank you.
Questions? Okay. All right, so let's talk about enzymes. So what type of molecules are enzymes? So what are they? Are they lipids, carbohydrates? Yes?
They're proteins.
Very good. They're proteins. Okay, so they have a three-dimensional structure like any other protein. And how do we disturb that three-dimensional structure? So if I want to take apart the structure of a protein, unfold it, or denature it, what can I do to it?
You use pH or temperature.
Very good. Excellent. pH or temperature, right? Just like any other protein. Okay, so enzymes have a particular pH where they work well at, and they have a particular temperature. You go above or below that, they don't work as well. Okay, so what else? Basically, what does an enzyme work on called? It's called a substrate, right? So enzymes are very specific for what they work on, right? So a nuclease works on nucleic acid. A lipase works on lipids, right? They're not interchangeable. Okay, so how does an enzyme speed up a chemical reaction, first of all?
It reduces the activation energy in the energy barrier.
Very good, excellent. Okay, so remember, every reaction has an energy hump to get over. That's called the activation energy or energy barrier. Okay, and so what an enzyme does is it reduces that hump. It doesn't get involved in the, it's not part of the product, but that's what it does. Okay, so now we're going to take some real world examples about enzymes and other things that can affect enzymes. Okay. Okay, so let's say this is the enzyme in 2D. Remember it has a 3D structure. That's my enzyme. Okay, all enzymes work on their substrate, so this is my substrate. So what's going to happen is the substrate is going to fit into a certain site on the enzyme, like a key fits into a lock. The enzyme's the lock, is the key. The keyhole is called the active site. So the substrate is going to hook onto the active site of the enzyme, and fit into the active site of the enzyme. Now, let's say I throw another molecule. Let's say I have both of these in a test tube. And let's say I throw something, I'm going to call it Y, that looks like the substrate. Would that speed up or slow down the reaction? So these keys look alike now.
Slow it down.
Okay, so slow it down. Why would it slow it down? You're right.
Because it's harder to tell which one should fit into that space.
You're right. So if Y gets in there, then the substrate's not going to get in there. Does that make sense? Okay, so you put the wrong key in to the lock, the right key is not going to go in. And so we have a problem there, right? So this is called competitive inhibition. We're going to take some real examples of this. Competitive inhibition. Inhibition just means blockage. Okay, why competitive? Because Y and the substrate are competing for the active site. Okay, and there are two kinds. There's irreversible and reversible. Okay, irreversible means the wrong key goes in and gets stuck in there, gets jammed. Reversible means the wrong key goes in but can pop back out. Okay, what we're going to do is we're going to take examples first of irreversible competitive inhibition. Okay. So this is irreversible competitive inhibition. And we're going to look first in bacteria. So what do bacteria have that animal cells don't have? Yeah, that's a bacteria. What do these guys have that animal cells do not have? Plant cells have it. Bacterial cells have it, and animal cells don't have it. Okay. What's this outer boundary of the bacteria called? You remember?
Cell wall.
Very good. Cell wall. So this is a cell wall. Okay. So it's critical to the bacteria for protection and other things. Okay. So what the bacteria does to make a cell wall is it takes these protein fibers, so that's the peptido part, and coats them with sugars. That's the glycan part. So these are like protein fibers that are sort of coated with sugars. Okay. Hold on 1 second. Okay, so what the bacteria does is it takes these protein fibers and uses an enzyme called transpeptidase. So how do I know this is an enzyme?
Because of the suffix.
Yeah, because of the suffix. Excellent. ASC. Awesome. Okay, so it takes this enzyme, what it does is it takes these protein fibers and sort of meshes them up, like so. So this is in fact the cell wall. So enzyme, product, substrate. That's the substrate. Okay, now what happens is I'm going to throw in a competitive inhibitor for this substrate. Okay, that's going to look like an upside down T or T, right? That means inhibitor. Okay, so the inhibitor are things like amoxicillin, penicillin, which I'm sure you guys have heard of, right? What these guys do is they block the enzyme by competitive inhibition, so the bacteria can't make its cell wall, the bacteria die. Any of the cillins do that, right? That's why they're pretty good antibiotics. We good? Questions?
No.
So the amoxicillin or penicillin would be the Y. Clear? Yes?
Yeah.
Yes.
Okay, awesome. All right, let's take a human example. Okay, we're still talking about irreversible competitive inhibition. Wrong key goes in, does not come back out. Okay, so this is in humans. And we're going to go into more detail in this when we talk about the nervous system. So there are nerve cells called neurons. Neurons are nerve cells. So they're really long cells. And they have a cell body, they have a long shaft over here. So the nerve signal is going to travel to the end of the shaft, right? And usually there's some other neuron or some other tissues. Let's say this is a muscle cell. Okay, the thing is, neurons never contact other neurons physically or tissues physically. There's always a gap between the neuron and whatever it contacts. Does anybody know what the gap is? Anybody heard of the gap? It's called a synapse. Synapse, out of biology, means gap. What happens is the nerve signal stops over here at the end, the terminal, and something's got to transport the nerve signal to the muscle cell. Okay, that something is called a neurotransmitter. Okay, so at the end of the course, we're gonna, towards the last month, we're gonna talk about human body and nervous system in detail. Okay, so the neurotransmitter is what transports the nerve signal to the other side. Okay, this one in particular for muscle cells is called acetylcholine. Okay, acetylcholine causes or delivers the signal to cause muscle contraction. Okay, now, let's say the neurotransmitter has done its job, acetylcholine has done its job, your muscle is contracted. Suppose, then we have to get rid of it. Suppose we don't get rid of it, what's going to happen to you? So let's say this neurotransmitter is left in the space here, keeps contacting the muscle cell, what's going to happen to you? Physically. Anybody know?
Is it going to be muscle cramps?
Sorry?
Muscle cramps?
Yeah, you're going to cramp, you're going to have spasms, more like, right? Eventually your muscles are going to keep contracting, they're going to die from overuse, right? So we have an enzyme called cholinesterase. Okay, that breaks down the acetylcholine and gets rid of it after it's done its job. So this one breaks down acetylcholine. Clear? Okay, so that's how we get rid of it. Now, if I block this enzyme competitively, you're going to have acetylcholine build up and you're going to die of spasms. The muscle's going to go into overuse. Some of the blockers of this enzyme competitively are nerve gas, like sarin. So nerve gas, it's reversible. You don't come back from nerve gas. But block the enzyme, this one builds up, and you get muscle contraction over muscle contraction. So have you guys heard of the insecticide raid? Right, those some of the insecticides you used to kill insects, yes?
Yeah.
Yeah.
So they basically contain something similar to nerve gas. They block insect cholinesterase, but on high enough levels, they'll start to work on you. Questions? We're good? Yes, no?
I have a question about muscle spasms.
Sure.
So technically they're caused because the acetylcholine technically doesn't leave the neuron?
There are various reasons. That could be one, right? I mean, and if that doesn't happen, that's quite a possible. Acetylcholine, well, a spasm or convulsion, more like a convulsion spasm occurs because there's too much acetylcholine there. One cause, right, of the spasm. Because the enzyme's not there to break it down. If the enzyme's not there, you're going to have too much acetylcholine and you go into spasms. Right? But that's not the only cause.
Okay, thank you.
Sure. Okay, so those are examples of irreversible competitive inhibition. Okay, what I'm going to talk about next are examples of reversible competitive inhibition. Okay, so in reversible competitive inhibition, the wrong key goes in to the lock, which is the active, to the keyhole, which is the active site, but can pop back out. Okay, so now we're talking about reversible. Let me write black. Reversible competitive inhibition, because we're still competing for the active site. Okay, so there's a protein in your blood called angiotensin 1. What's the name of the protein? It's about 10 amino acids long. AA means amino acids here. So it's a really small protein. It's converted by an enzyme called ACE. So this doesn't have a fancy name. It just means angiotensin converting enzyme into another enzyme called angiotensin 2. Angiotensin 1, angiotensin 2. This is only 8 amino acids long, so it's an even smaller protein. What angiotensin 2 does is it constricts blood vessels, tightens blood vessels, and causes an increase in blood pressure. So you guys know what hypertension is, right? Have you guys heard of hypertension? Right?
Yeah.
Yes.
Yes.
That means you have high pressure in your blood vessels, right? And remember, your blood vessels connect to organs. So if you have this high pressure blood heading to your organs, you can damage the organs. You can damage the tubes. There's a lot of pressure. That's why hypertension is bad. Okay, you can obviously die of it. So looking at this scenario over here, what causes a high blood pressure here? What could you do to reduce your high blood pressure? What could you block to reduce your high blood pressure?
Exercise?
No, here, just on this reaction.
Sorry.
No, but that's a valid thing. On this reaction, what would you do? What could you block here? Because I don't want my blood pressure to be lower.
You could block the AC.
Okay, I can block the ACE, which is what's often done, right? I block this enzyme, and I don't have this, and my blood pressure is going to go down. Now, why does it have to be reversible? Suppose it's irreversible, what would happen to you?
You would have too high of a blood pressure and ultimately like die.
Well, the problem is this, right? So remember what you just told me. Let's say I block ACE, right? So your blood pressure is going to fall, correct?
Yeah.
Okay. Well, if it's not reversible, if it gets stuck in there, that means your blood pressure is going to keep falling, right? And
Yeah.
you don't want that either, right? You don't want low blood pressure to collapse because of it. Okay, so some of the best antihypertensive drugs block ACE, the so-called ACE inhibitors. I'm just giving examples of some. Lisinopril, Vasotec, right? Cadopril. Okay, these are ACE blockers, and they lower your blood pressure, and they're reversible. After a while, blood pressure goes back up again, right? Able to reduce it to safe levels. Any questions? We're good? Yes?
Yes.
Yes.
Okay. So now, let's look at another example of reversible competitive inhibition. Okay, so let's say you drink some alcohol. Okay. There's an enzyme called ADH that converts this alcohol to something called acetaldehyde. Okay, acetaldehyde. Okay. You guys have heard of, has anybody heard of formaldehyde?
Yeah.
Okay, what's it used for? It's kind of noxious smelling. It's used as a preservative, right? So you've seen those animals in jars. So they preserve the organs of animals in jars, right? and it's kind of noxious and toxic. So acetaldehyde is sort of a cousin of formaldehyde, pretty noxious. So if this builds up in your body after you consume some alcohol, you feel very sick. Okay, so what your body does is quickly gets rid of this. Okay, so this is quickly converted by a second enzyme. So this is the first enzyme called acetaldehyde dehydrogenase. Okay, it's quickly converted into a weak acid called acetic acid. Okay, so that's what's happening here. Now, let's say I give you a chemical that blocks this enzyme. What's going to happen to you? The second enzyme. So what would happen to you? So I give you a chemical that takes out this enzyme. Reverse it, but I take it out. What's going to build up? Take a guess.
The amount of alcohol in your system.
Well, you remember what the alcohol is converted to? What's the alcohol converted to? That, right?
Right.
Okay, so if I block that, I'm not blocking this, I'm blocking this, right? If I block this, what's going to build up?
The acetaldehyde.
Aldehyde, right? This part here, and you're going to feel sick. Okay, so there's a chemical called disulfiram. Okay, that blocks this enzyme. But here's my second question. Who would I want to give this to make them feel sick? I'm going to block the enzyme. This is going to build up, and they're going to feel ill. So who would I give that enzyme to? I mean, this compound to? What kind of people? So somebody drinks alcohol. I give them this compound, blocks the second enzyme, this builds up, and they start feeling ill.
Maybe somebody that had alcohol poisoning.
But if they had alcohol poisoning, this is building up, right? So why would I make them even more sick? So I'm asking, what kind of patients would I give this to? So that when they drink alcohol, they feel bad. Or sick. This is more a conceptual, critical thinking question.
Patients.
So let me repeat it again. Who would I want to make feel sick after they drink alcohol?
Maybe like an alcoholic.
Yeah, absolutely. Right? Alcoholics, right? I mean, it's not a great way, right? It's like negative reinforcement, right? So this is sometimes called anti-abuse, trade name. It's sort of like a last ditch effort. You don't build resistance to it. The more you take, the stronger it gets. And it has to be reversible because you don't want them permanently nauseous. It's going to pop out of the active site of this enzyme. Clear? Any questions?
Yes.
We're good?
Yes.
Okay.
Yes.
All right. So now we're going to talk about a whole different kind of inhibition. Okay. So here's my enzyme. Again. Here's my substrate. So what's the keyhole the enzyme fits into? What's this part called?
The active site.
The active site. Great. Okay, now let's say I have an inhibitor. So let's say that's my inhibitor. My blocker. And let's say now you have to think of the enzyme in 3D. So let's say this is the enzyme. This is the inhibitor. So the inhibitor is going to hook up. Okay? So when that happens, this is what's going to happen. So from here, why would that cause the reaction to stop with the substrate? So why would the reaction stop?
Because it no longer fit.
Yeah, because I changed the shape of the active site, right? So there's no real world analog for this. So if this is the enzyme, this hooks up, maybe it does something like this to the enzyme. Okay? So this kind of blockage that you see here is called non-competitive inhibition. So why is it non-competitive?
Because no matter what, the substrate can't fit anymore.
Right. So we're not competing for what now?
For the keyhole.
Yeah, which is the active site, right? They're not competing for the active site. So again, you're going to have two kinds: irreversible reversible. Right? So irreversible means that doesn't pop back off. Reversible means it pops back off. Okay, so we're going to take some examples of this. Okay, first we're going to look at irreversible, non-competitive inhibition. Okay, so example one. There's an enzyme called ALAD. It's an abbreviation for an enzyme. And there's an enzyme called ferrochelatase. Okay, so this is enzyme one, this is enzyme two. Both of them are used to make hemoglobin. So remember, hemoglobin is an important protein in your blood that transports oxygen. Okay, so that's what both these enzymes help to do. Now, so let's say these enzymes are this. So there's an important blocker of both enzymes, and that is called lead. So lead, basically, it can affect you mentally, but can affect your blood by blocking the production of hemoglobin, by blocking these enzymes. Where can you get lead poisoning? Anybody know?
Paint.
Paint, exactly. You know, kids eat paint chips. And also lately, if you look at Flint, Michigan, the water, right? You get lead in the water. Okay, so this is a non-competitive, irreversible blocker, doesn't come back off. Okay, second example, yeah?
I can't read what that says right next to hemoglobin.
Make hemoglobin?
Next to the left of hemoglobin.
Oh, these two?
Yeah, I can't. I read the first one, just not the second.
Okay, so the second one is called ferro, F-E-R-R-O,
Mhm.
chelatase, C-H-E-L-A-T-A-S-E.
Thank you.
Sure. Okay, I'm going to give you a second example of irreversible non-competitive inhibition. Okay, so again, this is irreversible non-competitive inhibition. Okay, so example two. So there's an important enzyme in your brain and body called thioredoxin reductase. Okay, so this is a D, thioredoxin reductase, right? And it's an important antioxidant enzyme. So it gets rid of bad free radicals. Right? It gets rid of bad free radicals. Okay? Now, there's an important blocker of this enzyme, and that is mercury compounds. The compounds of mercury will block this enzyme and can be very lethal. Where can you get mercury poisoning?
Fish.
Yeah, fish, right? Okay. So about 20 years ago, 23 years ago, in the late 90s, 98, there was a chemistry professor at Dartmouth. Dartmouth is one of the Ivy Leagues on the East Coast. And she was working with mercury compounds. One of them was called diethyl mercury. And she spilled a few drops on the floor. Okay, and you go to clean, you put on your gloves, you take gloves and try to clean it up. The mercury penetrated her gloves and into her skin within 15 seconds. All right, and started doing this, and eventually caused brain damage. They couldn't get it out, and she died within 6 months. Okay, so some of these compounds are extremely lethal. Okay, any questions? We're good?
Yes.
Yes.
Okay, so now we're going to take an example of reversible, non-competitive inhibition. So in reversible non-competitive inhibition, this pops back off and the enzyme goes back to its original shape. Okay. So let's say we have, this is hypothetically, let's say we have A going to B, going to make ATP. Remember, ATP is the energy molecule of the cell. So enzyme 1 converts A to B. Enzyme 2 converts B to ATP. Assume that. So here's enzyme 1. Here's A. So here's enzyme 1. A is the substrate, and enzyme 1 is going to convert A to B. Sometimes in the body, we build up too much ATP. Okay? And when we have too much ATP, okay, that's a problem. It can cause problems, all kinds of reactions. So what's going to happen is ATP itself is going to, I'm going to draw it this way, hook up to enzyme 1, right? So it's going to hook up to enzyme 1 and change its shape. Right? So, you know, this is ATP hooking up. Okay, so it's going to change its shape. Okay, and basically now A can't fit into the active site. So what's going to happen to ATP production if A can't fit into the active site of 1?
It'll build up.
It'll go down, which is what we want. So basically ATP is blocking its own production. It's going to go down to safe levels, and then it's going to pop back off, and the enzyme is going to go back to its original shape.
So does the body do that to regulate it?
Yes, exactly. Are we good?
Yes.
Okay, so those are examples. Okay. All right, I'm going to give you one more example of inhibition that's different from competitive or non-competitive. Okay, and that'll be it for enzymes. Okay. So for that, I need to go back to the original energy chart. That's E. Here's my substrate. Here's my energy hump. Here's my product. So let's say the original energy hump was big. This is the enzyme hump. The enzyme reduces the activation energy. Okay. So the substrate goes from here all the way to the product. But over here at the top of this hump, it's neither substrate nor is it product. Okay. So over here, it's something in between. This is called the transition state. Okay. And we can model the transition state. Basically, the transition state is sort of a state of flux. Transition means from one to another. So it's not substrate, it's not product, something in between. Now, if we know what the transition state looks like, and we throw it into the enzyme reaction, what it does is it kills the enzyme reaction. But you have to know chemically what this in-between state looks like. Not easy to do. Okay? So this last class of blockers are called the transition state inhibitors. You're mimicking the transition state. Okay, so these are called the transition state inhibitors. Okay, I'm going to give you an example of one. So there's a virus called the influenza virus. What does the influenza virus cause?
The flu.
The flu. Okay, causes the flu. Okay, it has an enzyme called NEUR, N-E-U-R, aminidase. Okay, so this is an enzyme that belongs to this virus, and it helps the virus to spread. The enzyme helps the virus to spread from cell to cell. Now, if I look at this enzyme, this viral enzyme, and I know its substrate and know its product, and I sort of mimic the transition state, and if I throw it in there, create it in the lab, I can block this enzyme with my transition state inhibitor. And then I can block the spread of the virus. Okay, so they did that. They created a chemical called oseltamivir. Okay, so that's a transition state inhibitor. The trade name is called Tamiflu. Anybody heard of Tamiflu?
Yes.
Okay, so it's not the flu vaccine, it's a flu medicine, right? It blocks this enzyme. The virus can't spread. This is a transition state inhibitor, right? So it mimics this middle point here. Are we good? Questions? Yes?
No questions.
All right, so that is the material for exam one. Any questions for exam one? Okay, so whatever I go over from now on is going to be on exam two. So we're going to talk about cell respiration, which is how cells make energy, the second part of the lecture. And we will stop after that. Yeah?
Okay. Yeah.
Questions about the exam? Okay, so exam-wise, it's going to be like 35 or so multiple choice, 40, and a short essay, one short essay. You just have to answer. Yeah? Okay. So for the essay, you need to know what I just talked about. All the different enzyme inhibitors, how they work, and maybe one example of each. Yes?
Yes.
Yes.
Any questions on that? So now, that's what you should expect for the essay. Okay. Okay. So what I want to talk about next is something called cell respiration. Okay, so this is going to be an exam too, by the way, cell respiration, right? But you should know what cell respiration means, right? So what happens at the mitochondria is cell respiration. How cells make energy. Okay, so I'm just going to introduce it, then we'll take a break and come back and talk about the rest. Yeah. Okay, so basically we have electrons. Everything's going to revolve around electrons. Okay, so we have high energy electrons, we have low energy electrons. So this is what we're going to do. This is the gist of what we're going to do. We're going to take a high energy compound that has high energy electrons. We're going to extract those electrons away from that compound. And we're going to use these high energy electrons and make ATP out of it. We're going to use the energy in these electrons. Okay, that's our goal. So we're going to find some high energy molecule, take some electrons away. These electrons have really high energy. We're going to extract the energy from here and make ATP. That's our goal. So we need to know a few terms dealing with electrons. Okay. One such term is oxidation. Okay. Oxidation is loss of electrons. Okay, when something loses electrons, it is oxidized. Okay. 2, reduction. Gain of electrons. When something gains electrons, it is reduced. Okay, seems counterintuitive, but when something gains electrons, it is reduced. Okay. 3, anaerobic. What does anaerobic mean? Just the word. Anybody heard that word, anaerobic?
I think it's like without oxygen, I think.
Very good. Excellent, right? Without oxygen. Okay. And 4, aerobic with oxygen. Okay. So we need to know that term. So what we're going to do is we're going to break up cell respiration into 3 parts, 3 big parts. Most of it is going to occur in the mitochondria. The first part, which we'll talk about after the break, is called glycolysis. Okay, so glycolysis literally means breakdown of glucose. So glucose is my high energy molecule. That's what I'm going to use here. I'm going to extract high energy electrons from glucose. That's how we start. This is the only part of cell respiration, the main process, that occurs not in the mitochondria, occurs in the cytoplasm. Cytoplasm means the cell liquid. Clear? Okay, it does not occur in the mitochondria. We're good?
Yes.
Yeah.
Yes.
So here's the thing, okay? In your book, you're going to have many steps in each of these reactions, parts 1, 2, and 3. I just want you to know the important parts, what you put in, what you get out. You don't have to know all the reactions. It's not a biochemistry class. Okay? And so what we're going to start talking about after the break is basically what happens in glycolysis, what happens in the subsequent parts. Okay, and then we'll stop. Okay. One more thing for the lab, we're going to use the kind of virtual microscope. Did any of you receive the kit, by the way, from the vouchers?
Yes, I received it.
What's in
Yes,
it?
that's good.
What's in it?
A microscope, all the labs, a safety kit.
All of you guys received it? Yeah? No?
Mine's
I
on the
haven't
way.
received mine yet. Not yet.
Nawa, you said yes? No?
I said that it's on the way. They emailed me the tracking number.
Okay. Okay.
I have
All
a
right.
quick question
Yeah,
for
go
the
ahead.
people who received the tracking number. Are you able to go onto FedEx and track it? Because I pressed it and it says that it no longer exists.
Let
Not
me check
sure.
right now.
What happened?
I'll check.
Oh,
It's
Professor, no, it's because I tried to track it yesterday to
Yeah.
see where it was at.
Uh-huh.
And I pressed the tracking number for FedEx and
Yeah.
it said that it no longer exists. The status of that package.
Yeah, that doesn't make sense to me. Well, I haven't
Mine-
received that.
Mine showed that too, but then it came yesterday, so I think it's still gonna come.
Yeah, I think it's gonna come regardless.
yeah, mine says the same thing, but I'm just hoping.
Just wait on it maybe. Anyway, we'll use the virtual thing and I'll, so make sure that debugger is downloaded and stuff. And we'll talk about it. I have a little microscope here too, which I'll show you. That'll be okay. We're good? All right, let's take a break for about 30 minutes or so, and then we'll come back and talk about the rest. Yeah?
Yes.
Yeah.
Yeah. See you back here. Hey guys, you still there?
Yes.
Yes.
All right, let's start with glycolysis. Okay, so it's part one. Okay, so it occurs in the cytoplasm, not in the mitochondria. So we're going to start with a high energy molecule like glucose. And there's a whole chain of reactions which we're going to leave out. Okay. And so what we're going to do is we're going to extract electrons from this high energy molecule. So these are electrons that we're taking away from glucose. And now what we have to do is we have to have something to sort of hold on to these high energy electrons, right? You can't have them just floating around. So there's another molecule called NAD. This comes from a B vitamin called niacin. NAD is going to pick up the electron, hold on to it. It's going to pick up a proton to sort of balance the charge, and you're going to get NADH. So the purpose of NADH right now is to hold the high energy electrons till we're ready to use them. Okay? Now, as a side reaction, we're not going to make too much ATP. That's our goal, right? But we're going to make a tiny bit here. Okay, so we're going to take ADP, which stands for diphosphate, 2 phosphate groups, plus a phosphate gives you ATP. And the enzyme that does that is called a kinase. So here's the enzyme. So this is like a side reaction. Here's the enzyme, here are the substrates, and here's the product. Okay, so this side process here is called substrate. Okay, so there's my substrate, level, phosphorylation. Okay, substrate, and we're adding a phosphate group. That's where the phosphorylation comes from. Okay, now at the end of these chain of reactions, we're going to get a final product called pyruvate. Okay, so this is glycolysis in a nutshell. So what are the products of glycolysis? So what am I getting out of glycolysis here? What are my final products? Yeah? What are my final products? So what am I getting out of this? First part one.
Pyruvate.
Pyruvate. What else?
ATP.
ATP, right? What's the third one?
NADH.
NADH. Excellent. Okay, so this is what's holding on to the high energy electron. So NADH, pyruvate, ATP. We're good? Okay, so now we're not going to go to step 2 yet. Let's say we don't have any oxygen. Okay, let's say we're at a stage where we don't have oxygen. Now this could be, let's say you go to the gym, you exercise a lot, fast, and you run out of oxygen. Okay, what's going to build up in your muscles? Your muscles are going to get sore. What's building up? Does anybody know? Why does your muscle get sore? Okay.
Isn't it like lactic acid buildup?
Very good, right? So that's what's going to happen when you go on to anaerobic conditions, right? So these are, so I'm going to call this 1A. These are the anaerobic or fermentation pathways. So these are going to occur when you don't have oxygen, right? So let's say in your muscle cells that we just mentioned, right? That pyruvate is going to get converted to lactic acid by an enzyme called LDH. Okay? Lactic acid is sometimes also called lactate. Okay, and a side reaction, an important one, is that all that NADH we made gets reconverted back to NAD. Okay, this is an important reaction because in glycolysis, we're building up all this NADH. And if too much of it builds up with nowhere to go, like because we don't have oxygen, the entire process shuts down. And we don't want the entire process to shut down. So what we do is we recycle back your NADs. That's what the lactic acid pathway, fermentation pathways are for. Right? So we want to recycle NADs. That's the important part. Okay. Now, this is not the only anaerobic or fermentation reaction that occurs. There's another one, and that's in yeast. Right? So yeast do something similar. They take pyruvate, they convert it via an enzyme called ADH, but this time to ethanol. Okay, this is how you make alcohol or beer, wine, whatever. You put yeast under low oxygen conditions, anaerobic conditions, and you're going to get them to produce ethanol. The side reaction here is the same. NADH is going to get reconverted back to NAD. And you're also going to release a little bit of CO2. This is used to make bread rise. Right? So both these conditions are the anaerobic conditions, low oxygen conditions. Right? This is what happens. And the main purpose is to recycle back the NADs so you don't crash the system. Are we good?
Yeah.
So now, let's say we do have oxygen. We're going to go to step 2. And that is called Krebs or PCA cycle. And this occurs in the mitochondrial matrix. It occurs in the mitochondrial matrix. Right. So here's the mitochondria. It's got an outer membrane. It's got an inner membrane. This is the inner membrane. And the space between the two is called the matrix. So this is where Krebs cycle occurs. Again, there are a lot of reactions involved. I just want you to know what you put in and what comes out. Okay, so that pyruvate is converted into a molecule called acetyl CoA. Acetyl CoA enters a cycle which is Krebs cycle, and you're going to get some products. You're going to get CO2, so you're breathing out right now some CO2, and that's made in Krebs cycle. Now you're also going to get some NADH, more NADH. So does anybody remember what the function of NADH was? What did it do? What was the purpose of making NADH in the glycolysis? Yes?
Produces NAD.
Right, but why create it in the first place? What's happening? Why do we need it? Okay, because this is important to go to part 3, right? So we'll say it again. The purpose of NADH is to hold high energy electrons. It's an electron holder. Is that clear?
Yes.
Okay. So we're going to make a relative of NADH also called FADH2. This comes from another vitamin called riboflavin. This is an electron holder. This is also another electron holder, both electron holders. And we're going to make a tiny bit of ATP again, just like we did in glycolysis. That in essence is Krebs cycle. Okay, so what are the products of Krebs cycle? What are we getting out?
We get ATP, FADH2, NaOH, and CO2.
Yeah, sorry, this is a D, right? D
Yes.
and CO2. Are we good?
Yes.
Yeah, so these are the products of Krebs cycle. Okay. So now what we're going to do is we're going to go to part 3. Part 3 is the most complex. Okay, and that's called electron chain transport. Okay, and occurs on the inner mitochondrial membrane. Okay, so we're going to briefly describe that, or in detail actually. Okay, so here's my mitochondria again. Here's my inner membrane. And on that inner membrane are like 4 important protein complexes. These guys are huge protein complexes that are on the inner membrane. And they're going to function in electron chain transport. I'm going to give 4 a name. It's the only one I'm going to give a name. Here. Well, what's happening here is this. So here's my energy curve again. We've got these high energy electrons. Right now, they're being held by NADH and FADH2. Okay, what we're going to do now in electron chain transport is we're going to take those electrons away from these guys. We're going to oxidize them. So we're going to take the electrons, and that's what complex 1 does. We're going to take the electrons, and that's what complex 2 does. It's taking the electrons away. 1 and 2 are going to give up their electrons to 3. 3 is going to give up its electrons to 4. So what's happening here is we got high energy electrons here. They're being taken away by 1 and 2. 1 and 2 are going to dump into 3. 3 is going to dump into 4. And as we're going down here, down the staircase, we're going from high energy to lower energy to lower energy. And as we go from high energy to low energy, we're going to release some energy, right? If you go from high to low, you're going to release some energy, right? High energy to low energy. That's what's happening here. High energy to low energy, high energy to low energy, releasing some energy. 4 at the bottom of the staircase is going to give up its electrons to a molecule called oxygen. Oxygen picks up some protons that are floating around, and we finally get water. So you're breathing out not only CO2, you're also breathing out water, like when you fog up a glass plate, for example, or a mirror. Okay, so oxygen is your overall final acceptor of electrons at the bottom of the staircase. There is no other acceptor after oxygen. Okay, we're clear so far? Questions? Yes, no?
Yeah.
Okay, so keep this in mind. I'm going to leave this here for a second. Okay, I'm going to draw this inner membrane in a separate way. I'm going to draw it like this. So here's complex 1, here's complex 2, here's complex 3, here's complex 4. This is the matrix of the mitochondria. So NADH which we made in Krebs cycle glycolysis, is going to give up its electrons to 1. FADH2 is going to give up its electrons to 2. 1 and 2 are going to give up the electrons to who?
3.
Okay. We'll give up electrons to 3. 3 is going to give up electrons to who?
4.
4.
3. And 4 is going to give up its electron. What's the final acceptor? Who's 4 going to give up the electron to?
H2O.
Well, that's what we're going to make. But who's going to pick up the electron?
Oxygen.
There you go, right? Oxygen, the final acceptor. And we're going to get H2O, which you said. We're good so far? Yes, no?
Yes.
Okay. So keep this in mind. Okay? As we shuttle these electrons around, we're doing this, and we're releasing some energy. We're going to use this energy for something by the movement of electrons going down. Okay, so floating around in this matrix over here, mitochondrial matrix, are some protons, some H pluses. So complexes 1, 3, and 4 sort of double up as pumps, proton pumps. So what they're going to do is they're going to pump H plus from low concentration to high concentration. Does anybody know what low to high is? What's that movement called? We know high to low, diffusion. What's low to high? Does anybody remember? That you need to know for your first exam. Okay, remember active transport. Active transport. You guys remember that? Yes, no?
Yes. Yes.
Okay, so that's movement from low concentration to high concentration, but it requires energy.
Wait, could you maybe repeat that or write it down so I can
Sorry?
add it to my notes?
Sorry, what happened?
Could you repeat that or maybe write it down so I could add it?
Yeah, sure. So remember, active transport is mainly movement from low concentration to high concentration. So low H plus to high H plus, right? And it requires energy. Diffusion is the opposite, high to low. Yeah?
Yeah, thank you.
Sure. So what's going to happen is we need energy to do this. Okay, where does the energy come from? The energy comes from the fall of electrons. So as we're dropping the electrons from carrier to carrier, we're going to release some energy, and we're going to use this energy, or these pumps are, to pump out the protons. Okay, and there's a purpose to this. Okay, so it's also important that this membrane here, this inner mitochondrial membrane, is impermeable to protons. What does impermeable mean? What's the word?
Doesn't pass through.
Yeah, it cannot pass through. So what I want here, what the membrane wants here is, you pumped out all these protons, you don't want them to just flow back through. That defeats the purpose. They're building up here. They want to come in from high, just by diffusion, high to low. But they can't come in through the membrane because it's impermeable. We're good?
Yeah.
Yes.
Okay. So there's a fifth complex. Okay. This is actually another enzyme. This is called ATP synthase. Its job is to make ATP. This is where we're going to make the bulk of our ATP, most of our ATP. The only problem is it needs to be turned on, activated. The way it gets activated is these H pluses, which are building up, enter this enzyme and activate it. And when they do that, that's where we get the bulk of our ATP. Okay. So we're using these protons that are pumped out here they're going to enter the enzyme and they're going to make ATP. Now, this sort of subsection that I just described, this is called chemioosmosis. So chemioosmosis is like a subpart of electron chain transport. It's basically the pumping out of protons and the making of ATP, right? The job of these protons is to activate the enzyme. Are we good?
Yeah.
Yeah.
Okay. So here's my question. Let's say I give you a compound, a chemical, that blocks complex 4. It takes it completely out. Complex 4 is an enzyme. It's called cytochrome C oxidase. It's an enzyme. So it's one of the electron carriers. So I give you a chemical that just takes this out. What's going to happen to your energy production? ATP production. So you no longer have complex 4.
You wouldn't be making as much ATP.
Okay. Energy production will decline, right? Because you have no place to hand off the electrons to and enough energy to generate this movement. Yeah, that's what cyanide does. You guys know what cyanide? You guys have heard of cyanide? Yes? You ever heard of cyanide?
It's the poison that soldiers would.
Yeah, people commit suicide with cyanide, right? Because it's kind of quick acting within a few minutes. It's also used in executions. It's also used, in that's by gas chamber, right? You basically use cyanide gas and you execute people. Why? Because it blocks complex 4 and shuts down ATP production, energy production. And your cells die and you die quickly. Are we good or not?
Yeah.
Good.
Okay. So let's go to another question. And after this, we're going to talk about photosynthesis a little bit and stop. Okay. Let's say in the 30s, let's write this down. Okay, so let's cyanide blocks complex 4 and stops ATP. Okay. So in the 1930s, there was a diet pill called dinitrophenol, DNP. Okay. And what it did was it basically took these protons and it could dissolve itself in lipids. It was lipid soluble, which these membranes are, right? It's like a membrane. It took these protons and brought them right back down. So I remember I said this was impermeable before. Well, what this chemical did was it took these protons and made this membrane sort of leaky to protons and brought them right back down. What's that going to do to energy production? Or ATP production?
Stop it.
Sorry?
For the energy or the ATP production, would it like block it or?
Yeah. What? Yeah, you're right. Why would it stop it? So if that chemical took all these protons and brought them right back to the membrane.
Because then they aren't going to cause activation for the ATP synthase.
Excellent. Right. It can't, and she's right? You can't cause activation of the ATP synthase, right? Because they got to cause activation. So remember, we started with glucose. All of this started with glucose. So you can't make any energy from glucose if I give you this chemical. So can somebody tell me what will you try? What's the other source of energy in your body? What will you try to burn off?
Lipids?
Yeah. So you start burning off fats, right? That's why it's called a diet pill. There's only one problem. If you start burning off fats really fast, which is what this chemical does, your body temperature starts to rise. So your body temperature starts to go up to 110 degrees. Okay, so what happens to you at 110 degrees? So what's going to happen to your body at 110 degrees?
You start dehydrating, so your cells start like shriveling again.
Well, let me put it this way. What happens to your body at 104 degrees, 102 degrees?
You start to sweat.
You sorry?
Sweating.
Well, what's 102? 102 is a fever. How do you feel?
Fever. Yeah.
Do you feel good?
No.
Yeah, because your enzymes are not working as well at that high temperature, right? Now, increase that temperature to 105 degrees. How are you going to feel?
Terrible.
Okay, now increase it to 110 degrees. What's going to happen to you?
You could die.
Yeah, you die, right? Your body started cooking, right? And so that's what DNP did. It caused deaths that way, right? So I'm going to show you something. So it's still, by the way, you can still get this pill on the internet, right? Especially in England, right? So these are all the DNP. This was a few years ago, used by slimmers and bodybuilders, right? She was a student, university student. She was a med student, and these people all died, right? Because of you can get it on the internet, right? Make sense?
Yeah.
Yeah.
Yes.
I have a question.
Sure.
So would those high temperatures basically unravel the proteins, or would they need to be a lot higher?
No, you'll start to mess them up,
Okay, thank you.
and then you'll just cause organ damage, and I guess that's what happened to these kids, right?
Yeah.
It was about a year ago too, right? I mean, it has legitimate uses, but people are not careful. It's like anything else. You gotta be careful, right? The DNP makes mitochondrial, but everybody's into weight loss, right? So it's a big draw, right? Cool. Yes. So that's it for cell respiration. I'm going to introduce the topic of photosynthesis and we'll stop. Okay. So a lot of this stuff, like cell respiration, photosynthesis, we need to do this stuff. I mean, it's not of great interest to me, but it's part of the course, right? The next part, which is DNA, gene engineering, that's the cool stuff, at least to me, right? I'm biased, right? There's a company called Colossal, just received $15 million to clone a mammoth, okay, using CRISPR technology, which we'll talk about in next week or the week after, if anything, right? But that to me, I mean, people don't like it and stuff, but it's pretty cool because you can engineer elephant DNA and introduce mammoth sequence of DNA in there and create a mammoth. They just received $15 million, right? So we'll see what happens. Make sense?
Yes.
So we shall see, my friends. Okay. So photosynthesis. Okay, so photosynthesis okay, so the first plants that were photosynthetic, that means the usage of light, they were not plants. They were purple bacteria. And they use a compound. So this is about 2 billion You kids are good with acronyms. This means 2 billion years ago. Okay. So hydrogen sulfide gas, it's a poisonous gas, some CO2. They used some light energy. That's where the photo part comes in. They made glucose for themselves, and the waste product was sulfur. Okay. Now, around 1 BYA, billion years ago, another group of bacteria called the cyanobacteria okay, they use a different compound. They use, instead of H2S, they use H2O, water. They use CO2. They use light energy. They made glucose, just like these guys, but the waste product wasn't sulfur, it was oxygen. So the release of oxygen into the atmosphere had implications for all of us. Right? So ocean plankton, ocean green stuff is important to us. So that's where most of our oxygen comes from. Okay, so plants are pretty cool in that what they do is they take water and they actually split the molecule apart, not evaporate it, but split it apart, literally, chemically. Very tough to do in a lab, but a plant can easily do that. But we're going to look at this process in detail, how a plant splits water apart. So for now, okay, one last thing on electron chain transport. That's where 95% of the ATP comes from, right? All of our ATP is made there, or most of it. Okay, so let's talk about light. Is light just one color? What are the colors of light?
It's a whole spectrum.
Okay, it's the whole spectrum, right? So red, orange, yellow, green, blue, indigo, violet. So that's the visible spectrum. You can't see outside. So here would be ultraviolet, here would be infrared. By the way, birds have more cones in their cells, their neurons, than us, and cones perceive color. So birds can see into the ultraviolet and other light spectrum. They can see 1 million times more color than you do. Right? Certain shrimp, like the mantis shrimp, have like 12 cones. They can see even much more than birds. Okay? So here's the different wavelengths or colors of light. We need something to absorb that. Okay, and that something in plants is a pigment called chlorophyll. Right, so chlorophyll is a pigment that absorbs light. Okay, so if I were to plot the absorbance, so ABS means absorbance of chlorophyll, and the different lights here from violet to red, I'll find that chlorophyll absorbs mainly in the blue-violet and red-orange range. Where it doesn't absorb well is like in green, right? So plants, a lot of plants appear green to us. Why? Because that's not absorbed by the plants, reflected back to your eyes. Right? The plant has other pigments other than chlorophyll to absorb other wavelengths, other colors of light. But this in general, blue, violet, red, orange is what it likes. Clear?
Yeah.
Yes.
Okay. So the organelle where this happens, which is the last thing we'll say, is the chloroplast. So remember, chloroplasts have their own DNA, just like mitochondria. So here's my chloroplast. Inside the chloroplast are these sacs. These sacs are called the thylakoids. Okay, in these sacs, in these sacs is the pigment chlorophyll. Outside the chloroplast, this is called the stroma. So what we're going to do next week is we're going to break up photosynthesis into two parts: light-dependent reactions, light-independent reactions. So where in the chloroplast do you think light-dependent reactions are going to take place? Which part?
The thylakoids?
Yeah, why? You're right. So why are they going to take place in here? The light-dependent ones.
That's where the chlorophyll is.
Very good, right? So remember, chlorophyll is the light-absorbing pigment. It's in here. So that's where the light-dependent reactions are going to occur. Are we good?
Yes.
How are you guys doing? You all right?
Yeah.
Yeah.
Good.
All right. So let's stop here. We'll pick it up next week. Any questions on the exam?
Is the exam just what we've been through up until now?
Not cell respiration and photosynthesis.
Okay.
Don't stop at enzymes. Right? Cell respiration and photosynthesis we'll talk about in, we'll do it in the second exam. We're good. Other than that, no other questions exam-wise. So on Zoom next week at 8 a.m. with cameras on.
Are you going to want to see the paper that we're writing on or
No,
face?
no. Is he going to submit it online?
Is it open note or no?
No.
Okay.
And you have 1 hour too. 8 to 9. Yes?
Is there a study guide or just our notes?
No, there's study guide too up there. You guys find it? You guys know where it is? Yes, no?
Could you show us where the study guide is, please?
Of course.
Thank you.
You guys know where to find my recordings too, right?
Yeah, and Confer Now.
Yeah. And some people are not sure. All right. So you go into files, click on files. And when you click on files, you'll see all the files, right? And so go down here to study guide. All of them are here. Study guide exam 1, 2, 3, 4, 5. Yeah?
Yes.
So what was the essay on? You remember?
It's on enzymes.
Yeah. How they work and the inhibitors that we talked about, right? Competitive, non-competitive. You have to give every example, one per group, transition state. Yeah?
Yes.
Okay, and remember to take your lab quiz, okay? 11 and on pH, 11 and 12.
Yeah.
All right, so we'll meet back here at 12 and talk about the lab, yeah?
Yeah. Thank you.
All right, we'll see you soon, guys.
Thank you, Professor.
Thank you.
Thank you.
Recording stopped.
I have a question.
Tell me.
I don't think, are you planning on posting the lab quiz later? Because I don't think it's posted.
Should be. Let me see. Let me see. So go to modules. This is the pH quiz. Okay, so I think you need to just refresh it. It's there. The pH quiz too.
Oh, okay, perfect. It was just not on the calendar, that's why.
Yeah, it's there. Oops. Yeah.
Thank you so much. See you back at 12.
Yeah. Take care.
Recording in progress.